Thank you and welcome to today's talk on the hydrodynamic characteristics of juvenile unionid mussels. My name is Joe Ackerman. I'm giving this talk on behalf of Julian, who received his master's a few months ago. He's now working in the hospital in Orangeville, helping us in these trying times. I'd like to begin by thanking the organizers for getting this conference up online. This is a great opportunity for all of us. And I'd like to thank my funders, especially the Canadian Freshwater Species at Risk Research Network, funded by Fisheries and Oceans Canada. Unionid mussels are cosmopolitan freshwater ecosystem engineers, largely because of their suspension feeding and burrowing in river bottoms. Unfortunately, about 75% of the species, and there are about 200 of them in North America, are at risk, largely due to habitat loss, land use changes, introduced species, and the fact that their larvae are parasites on vertebrates, mostly fish, and so anything that affects the host therefore affects them. Here's a life cycle you can see here on the bottom right. Sperm is broadcast, taken up by females, that fertilization occurs, larvae are brooded, then released, and they parasitize fish. When the larvae metamorphose into juveniles, they detach or exist and settle down to the river bottom. They're fairly small when they exist, and we don't know where they settle into the river bottom or where they are successfully settling into the river bottom. So this talk is about where juvenile mussels exist in the riverbed, and we're trying to see if hydrodynamics such as bed shear stress affect where they are and therefore where they can be found, so we could then learn more about their habitats. Here's a juvenile sitting on the bottom. It lives in a velocity gradient. The arrow indicates the magnitude of the water velocity, and because of this they experience drag downstream, lift away from the surface, and of course they can be flipped over by this gradient in velocity. That's the shear stress, which is a force that's acting normal to the mussel, and it's divided by unit area. So these cause things to move in bottoms. Shear stress sub C is the threshold of particle motion, and we measured this at 0.2 pascals in the laboratory. This was work based on previous graduate student Sarah French. In rivers, there's a lot of variability longitudinally at small scales because of the river pool sequence. So here you can see riffles and pools, and there are elevational changes, changes in slope that lead to differences in water velocity and depth, and of course these differences in shear stress. And we're quite interested in how such changes might influence the settlement and persistence of these juvenile unionids, which we defined as less than 30 millimeters in length. And previous studies have found that juveniles can be found behind boulders and within river pool sequences, and also in riverbanks. But we don't have a lot of information on the physical conditions that exist in those areas, and that's one of the reasons for this study. We looked at a river reach in the Sydenham River in southwestern Ontario. Near Florence, it has a high mussel density of about 24 mussels per meter squared, and there are about 24 species present in this one reach. Riverbed was mapped at 50 by 50 centimeter resolution. We used a GNSS system, which connects to the satellite to get accurate within 1 centimeter bed elevation, and also an acoustic Doppler velocimeter to measure velocity. We surveyed a number of conditions using these techniques. We calculated shear stress based on two different approaches. First, a large spatial scale approach using the depth shear product, where the shear stress is a function of density, gravity, the water height, and the slope along the reach. It's a broad scale measurement of the total shear stress across the reach. We also measured local shear stress at individual locations using the law of the wall, which was applied to velocity profiles taken at those locations. So here you can see a velocity profile, height above the bed and the velocity increasing. And you can see it has this kind of a logarithmic portion, especially near the base, and that's where we want the measurements to be made. And we used a nonlinear least squared fit to determine the shear velocity U star, and then based on some other relationships, could figure out the local shear stress. Quadrats were placed approximately every 1 meter along transects, and we also measured the pore water chemistry. The transects were then excavated using an airlift system, which is shown here in the image, and also by hand for larger material we could pick up. And this material was passed through a series of sieves to find out the relative contribution of different sizes. And the top 3 sieves were examined for juveniles because they would likely be present in those size ranges. We use logistic regression to the presence-absence of juveniles. So this technique gives us a probability of finding juveniles based on model inputs. We use stepwise selection, AIC, residual deviance, and the fit to the data to choose the appropriate model. Principal component analysis was also used to characterize the juvenile habitats based on the material the variables I just presented. What does the data look like? Well, here's the river reach flowing from top right to bottom left. The arrows indicate the relative speed and direction, and the larger the arrow, the faster the speed. So there's quite fast speed along the eastern bank, a little bit slower here on the western bank. Here's a location of high shear stress transects, low shear stress transects based on the depth slope product, and then some cross-stream transects. This other map shows the bed material, boulders, cobbles, pebbles, sand, silt, etc. And you can see that the circles here are areas where we found no mussels. X's would be where we found adults, and the solid where we found juveniles. So adults seem to be found throughout the reach, but juveniles are located mostly on this western bank. The local shear stress, you can see here the relationship between the observed, the red velocity profiles, and then the fit to that law of the wall. So the data show quite a nice correspondence to the model. Here is the frequency distribution of the 118 quadrats which we sampled, and this is the shear stresses that we observed in those quadrats. We found quite a number of juveniles. They're presented in the red bars, and then the blue are the adults. As you can see, the red bars, there were about 5 known species. 4 of them are presented here on the bottom with, you can see these are millimeter increments on a ruler, and a number of individuals were too small to use diagnostic tests to identify them. So all of our mussels were returned to the field along with sediments, so we would not be disrupting their habitats. There was an association between shear stress and coarse sediments. You can see here that mussels, the juveniles, tended to be found in locations that had large amounts of sand, but also in areas that had lower shear stress. When we plot those together, here's local shear stress, here's the sieve size. You can see that there's a narrow band of shear stresses that they're found in, and a much wider band in terms of mass of 2,000 micrometer sieve. This is a distribution of local shear stress. The red indicates the number of individuals, the proportion of individuals that were found, and the black represents the logistic regression model, the best one. It matches quite nicely. The peak is a little bit offset, but this model, as you can see, uses shear stress and shear stress squared. The first was marginally significant, the second was statistically significant, and we only used 2 predictor variables because we had limited sample size, so hence that limitation. Clearly, these juveniles were found at much higher shear stress than what we predicted from the laboratory results. The mean was around 0.52 and the maximum was about 0.77. In terms of the habitats, this is a PCA. You can see strong in a representation of dissolved oxygen. There's shear stress, the mass of the 2000 sieve, water temperature, specific conductivity. So you can see the first two axes present explain quite a bit of the variation. The different shapes represent different species. You can see here, interestingly, that the 5 spike we found are mostly in this northeast quadrant, and the northern riffle shell tend to be to the left of 0 on PCA 1. I did mention here that the different colors indicate the relative size of those individuals. Similar pattern now for adults, although there's a much larger range of sizes and more species that were found. Interesting though that the spike tend to be found in a much broader range of conditions, as do the northern riffle shell. So juveniles seem to be much more restricted compared to adults, even of the same species. So we found significant differences in these characteristics in quadrats with and without juvenile mussels. Logistic regression identified a significant nonlinear relationship, which will help us locate juveniles in the field. Shear stress has been predicted as a key factor for juvenile habitat, and this is based on inferences from field excavation and large-scale hydrodynamic models. Our results, 0.77 pascals, were larger than the predictions of 0.26 from the lab, and that's largely due to the fact that we have larger sized individuals in the field, more variable riverbeds, and the fact that there's quite a bit of mussel burrowing, so there's behavioral implications that could affect the results. Juvenile habitats, 0.3 to 0.5 pascals is the area to find the highest probability of juveniles, but sediments are also important components. They seem to be found in areas that are more depositional, containing coarser material, coarser sandy material relative to the rest of the channel. We didn't find juveniles when the shear stress was too low, perhaps because they're buried by silts under those conditions, or when the shear stress is too high, when they potentially are scoured from the bed by the hydrodynamic forces. 3 environmental variables, dissolved oxygen, pH, and temperature, loaded highly on PCA axis 1 and 2, highlighting the importance of pore water chemistry, which was recently also reported by Fung and Ackerman in a different state. There are some implications for mussel conservation. First, we must protect and restore natural environmental flows to maintain the shear stress conditions and sediment conditions for juvenile mussels. In other words, we must maintain stream integrity in natural flows. Even with the small sample sizes presented here, there seem to be habitat characteristics that differ among the several species that we observed, which suggests that maybe species-specific research and recovery actions might be needed. One size doesn't fit all, in other words. Our results will help inform future conservation efforts directed at approaches like reintroducing artificially reared juveniles to nature. We'll have a better idea of where to place them based on this research. And if you'd like additional information or any information about what goes on in my lab, please have a look at ackerman.uoguelph.ca. I thank you so much for listening to this talk, and hope you have a great day.